I request you all to please take a seat and kindly switch off your mobile phones or put them on the silence mode. And upon request from Professor Varmus, it would be best if you do not come very close to the stage clicking photographs. You may click from your seats. That would be easy on him. Now I welcome you all to the 54th Meghnad Saha Memorial Lecture on behalf of the Saha Institute of Nuclear Physics. This year, we are celebrating the 125th birth anniversary of Professor Meghnad Saha. As a part of that celebration, throughout the year, starting from the 6th of October this year, we will have series of scientific meetings, conferences, workshops. This lecture, the M.N. Saha Memorial Lecture, happens to fall at an opportune time. This lecture series has been delivered by stalwarts like Niels Bohr, like Hargobind Khurana, and many others, who have done exemplary work in the fields of physics, biology, and chemistry. Today, it is an honor for us to have Professor Eliot Varmus amidst us delivering this lecture. Now, I request our director, Professor Ajit Kumar Mohanty, to please come on stage and introduce the speaker.
Good afternoon to all of you. Honorable Professor Eliot Varmus, respected dignitaries, colleagues, invited from the media, students, ladies and gentlemen. Professor Eliot Varmus, Speaker of the Day, doesn't require any introduction in any parts of the world. However, being customary, it is my pleasant duty to introduce the 54th Meghnad Saha Memorial Lecturer, Professor Eliot Varmus, the co-recipient of the 1989 Nobel Prize in Physiology or in Medicine for studies of the genetic basis of cancer. Professor Varmus joined the Mayer Cancer Center of Weill Cornell Medicine as the Louis Thomas University Professor of Medicine in April 2015. He is also a senior associate member of the New York Genome Center, where he helps to develop programs in cancer genomics. Previously, Dr. Varmus was the director of the National Cancer Institute for 5 years, the president of the Memorial Sloan Kettering Cancer Center for 10 years, and director of the National Institute of Health for 6 years. A graduate of Amherst College and Harvard University in English Literature and of Columbia University in Medicine, he was further trained at Columbia University Medical Center, the National Institute of Health, and the University of California, San Francisco, before becoming a member of the UCSF. Basic Science faculty for over 2 decades. He is a member of the USA National Academies of Sciences and Medicine. He is involved in several initiatives to promote science and health in developing countries and serve on advisory groups of several academic, governmental, philanthropic, and commercial institutions. These positions currently include co-chair of the Mayor's Life Science NYC initiative and member of advisory boards for Chen Zuckerberg Science, the Board and Crick Institute, and 3 biotechnological companies. The author of about 400 scientific papers and 5 books, including a 2009 memoir entitled The Art and Politics of Science. Varmus was the co-chair of President Obama's Council of Advisors on Science and Technology, a co-founder and chairman of the Board of Public Library of Science, and chair of the Scientific Board of Gates Foundation, Grand Challenge in Global Health. And the introduction will go on soon, but with this brief, I'll end it here and invite Professor Varmus to give the 54th Meghnad Saha Memorial Lectures.
Okay, thank you very much. I didn't mean to expose you to the secrets of my desktop display of files, but we'll see if we can get things back on track here. Good, okay. Now I'm sorry the screen is so far away from you, but I think there's no opportunity to redesign this auditorium today. So instead, we're just going to move ahead and hope that even those of you in the back row can see something that's on the screen here. It's a real pleasure for me to be invited to give this talk. I'm a great admirer of the person for whom this wonderful establishment was named, and I congratulate him on reaching 125 years after his birth. And it was a real treat and an honor to be able to place a chain of flowers around the statue that you see at the entry to this institute. This is a memorial lecture. It's a celebration, and I'm going to use it myself, rather selfishly, to talk about some memories of my own, because around 50 years ago, what for many people in this audience, the students, might seem like a very long time, about 50 years ago, I came to India for the first time to travel to a place where I was going to work in Uttar Pradesh, and I had a very profound experience in Calcutta, as we then called it, and I want to spend a couple of minutes at the very beginning to reflect on those first experiences in Calcutta, because they turned out to have been the first moments in a series of life-changing events that occurred over the next few years that allowed me to set a direction that resulted in a lifetime engagement in efforts to understand the genetic basis of cancer. And so I'll spend a few minutes deep in nostalgia about those 4 years, which occurred around 50 years ago. Then I'm going to spend some time talking about the situation in cancer research at the time that I and my colleagues got involved. I will say something about the work that we did in the early days, mainly in the 1970s, and I will tell you about how that work that was carried out with viruses of chickens that had the ability to induce cancers in animals turned into an effort to understand how cancer arises using tools that may seem fairly primitive by today's standards. We didn't have recombinant DNA technologies, we didn't have high-throughput sequencing, we didn't have cryo-EM, we didn't have the human genome to study, but it was still possible to begin to identify the precise genes that play a role after they are mutated in generating cancers, and not just allowing us to understand cancer better, but actually to treat and possibly to prevent it more effectively. Then I want to spend some time at the end talking about some social movements that have been affected by the changes that have occurred in cancer research. A greater attention to cancer in the developing world, and some reflections on what is required for successful pursuit of a scientific career in this current age. And then we'll be free to go home. So, as I say, I want to spend a few minutes speaking about things that happened in relatively quick succession in my life around 50 years ago. The first thing was actually arriving in Calcutta in March of 1966. I landed at what we then called Dum Dum Airport. Hope you can see that. And to my surprise, not having been in India before, the city was in an uproar. We arrived at night. We drove into the city with thousands of soldiers with poised rifles guarding the road. The next morning when I woke up, I found there were crowds in the streets. Buses were being overturned by angry students. And indeed, it wasn't until many years later when the American Indian novelist Jhampira Lahiri wrote a book called The Lowlands, which I highly recommend to you, that explained what was happening politically to Calcutta at that time. Parenthetically, one of the people who encouraged me to go to Calcutta was a college friend named Leonard Gordon, who had written a book about the role of the Bose brothers in shaping Bengal and India's attitude toward the Second World War. That's a much longer story. At the same time that I was looking out the window, or looking out from the top of certain buildings I was visiting at the turmoil in the streets, I was also seeing some pretty remarkable things. I had a chance to visit the Johns Hopkins Center for Cholera Research, where oral glucose treatments were being developed for the treatment of cholera, and learned about the work that was being done by Indian and American scientists at that place. Because of my connections through my friend Leonard Gordon, I was able to meet with the renowned film director, Satyajit Ray, in his house for an evening and talk about his history in the film. And actually, I was delighted this morning to see in the Times of India a long story related to this year's Calcutta Film Festival, I should say Kolkata's Film Festival, about the legacy of Satyajit Ray and the efforts that are being made now to restore some of the films of his which have undergone a fair amount of decay in storage over the last some years. Now, after my several days in Calcutta, I then set forth up the Ganges, not down the Ganges, to the place where I was going to work for the next several months, the Clara Swain Mission Hospital in Bareilly, UP. Some of you may know that as the place where you get off the train to get on a bus for Nainital, but it's also the site at which the first Methodist mission hospital was founded in Asia. It was a hospital staffed by very well-trained Indian physicians as well as a number of Christian missionaries. I am not a religious person. I was just there because I was looking for some place to use a fellowship that would bring me into contact with patients with diseases I was unlikely to see in New York, where I was working as a medical student. This was a place to test out my interest in having a career in what we then called tropical medicine. And I saw many profound cases of patients with tuberculosis and leprosy and other infectious diseases. I was impressed by many aspects of the way this hospital operated, but I also found that practicing medicine was probably not going to be the course I would take for the rest of my life, that I was more interested in knowing why these patients were sick, how we could develop better ways to treat them, and I was realizing that I was going to have a life based in American academia, more likely than I would in a hospital in India or other developing countries. Now, about the same time, just a couple of years later, I first began to do some serious science. And the reason, one of the reasons that happened was because I did have some proclivity toward working in academia. Some research credentials were required, but I also was, like most of my colleagues, required, because of America's involvement in the Vietnam War, a war with which I had no sympathy, to either leave the country, serve in the Army, which I was not prepared to do, move to Canada, go to jail, or work in the public health service. And many of my colleagues shown here were, like me, spending a couple of years after medical school and a couple of years of training in clinical medicine at the National Institutes of Health, where the Public Health Service had one of its workstations. That's me. And one of the things that's very striking about this picture, especially striking, I'm sure, to the women in the audience, is that there was a bit of a gender imbalance here. Virtually everybody who was in the Public Health Service at that time, working as trainees at the NIH, was male. There were a couple of reasons for that. One is that only 5 to 10% of all medical students in the US at that time were women. That, I'm glad to say, has radically changed. And secondly, women were not required to serve in the Army during the Vietnam War, whereas males were. Now, during that period, as I say, I became familiar with laboratory science. My mentor in that endeavor, Ira Paston, is shown on your left, speaking at my 60th birthday party with a close colleague of mine, David Baltimore, who won a Nobel Prize for discovering reverse transcriptase, the viral enzyme that drives replication of retroviruses, a class of viruses we're going to spend a lot more time talking about in just a moment. So the unusual part of the story in some ways was that I was finally doing some science. I was 28 years old. This certainly in India, where you start making your career choices very early, and even in the US, it's virtually unheard of these days for someone to begin to do science at that age. Before that, in addition to doing my 4 years of medical school, I had been a student of English literature, receiving a degree in literature both from my college, Amherst College, and Harvard University. But the American educational system is happily quite forgiving. One can have what I like to call a prolonged adolescence and make very late changes and go from literature to medicine to science over the course of less than a decade. Now, my time at the NIH, working in Ira Paston's lab, was very important for subsequent work that I was able to do in cancer research. And without taking you through the actual experiments I was carrying out, I just would like to convey a few of the messages that I got from work that I did in his lab, trying to understand how cyclic AMP regulated gene expression in E. coli, a commonly used bacteria. The first message, and the most important, is that very simple models studying one well-studied gene, the lac operon, in a simple organism, a bacterium, would be very useful and prove to be very useful for understanding more complex signaling events that might occur in much more complex cells, like cells of human beings and other mammals. Secondly, I learned that progress in trying to understand biological phenomenon, even in these single systems, was very dependent on certain technical tools, like having good mutants, good genetic tools, or having the ability to look at and measure the affinity of two pieces of nucleic acid for others, a method called molecular hybridization that allowed measurements of the presence and quantity of, for example, messenger RNAs. I also learned that there is no end to science, that if you got a clear answer to a clear question, you had more questions. Some of these would be relatively uninteresting, but many of them would be profound, and that science was not a matter of absorbing things in textbooks. It was a matter of asking a good question about something we don't know, trying to answer it, not simply from the perspective of getting an answer, but to allow yourself to then ask even more profound questions about how nature works. I also learned about the pleasure of telling other people about what you've found, and that was normally exercised through the publishing process. And at the end of this talk, I'm going to say a few things about publishing in the modern era and how we get word of our accomplishments and even our disappointments to our peers and to our competitors. That is a very important part of doing science, sharing our knowledge, reacting to the knowledge generated by others, and it's one of the things that has motivated many of my own activities that were political in nature over the last 10 or 20 years. And finally, I learned by taking courses at night at the NIH that major problems in the causation and mechanism of disease might now be amenable to dissection as a result of advances in molecular biology that were allowing us to measure proteins and their concentration, to look at messenger RNAs and the control of transcription from complex genomes by using some of the methods that had been made available. Now, my wife would be very disappointed if I didn't point out that those few years just after my time in India included an important event, our marriage. This is a picture not from our marriage, but from a sabbatical we took about 10 years later in London with our then very young children. You would not recognize them today from these pictures, since they're now in their 40s. Nevertheless, that was an important event that shaped many aspects of my life. And you can tell my wife I did tell you all about that. Now, at about the same time, while I was, just after I got married and was starting to think about what happens after the NIH, I began to think about what I would do next, and I had already decided that my life was not going to be shaped around practicing medicine and curing or not curing patients, but was going to be devoted to some aspect of the use of molecular biology to study an important disease. I had in fact learned very little about cancer at my medical school. My medical school was famous for, in many ways, for good things, but also famous for basically ignoring cancer as a problem too hard to solve. And hence, I really knew very little about what was going on in the world of cancer research until I was at the NIH and took an evening course on mechanisms of carcinogenesis and learned that while there were some clues to the idea that genes were important in cancer, I'll review those clues in just a moment, that in fact there really weren't experimental systems at that point that were extremely attractive. There were reasons to believe that genes were important, but nobody had any idea about what specific genes were actually candidates for being involved in the generation of a cancer. Indeed, we knew about very few genes. We knew there were proteins, and we thought proteins are probably encoded by genes. There had been some study of globin as a gene that is mutated to generate diseases like thalassemia and sickle cell disease. But basically, this was pretty much a black box. And during my courses at the NIH, I learned that there were a few people, especially in California, who were trying to use genetically simple organisms, viruses, both viruses with DNA as their genome and viruses with RNA as their genome, that were known to cause various kinds of cancer in animals. That seemed like an entry point to try to get some handle on the nature of the genes that actually might be responsible for human cancers. So I went out to California, I shopped around, met some people I didn't think I'd get along with, and then suddenly one afternoon, unexpectedly, I ran into this guy, J. Michael Bishop, who became my scientific partner in many ways, and you'll see some evidence of that later on. And we decided that we would do some work together to try to understand properties of one important member of a virus class called the retroviruses. You all know what retroviruses are because HIV is a retrovirus. Until that came along, often people were rather confused about what retroviruses might be. And I will come back and tell you more about them in just a moment. But at about in that period, around the late 60s, early 70s, other things were happening, partly as a consequence of what had happened in the 1950s, creating times that now to current graduate students and medical students and postdocs in the US, when work seems very stressful and hyper-competitive, looking back, things looked a lot easier with respect to simply getting involved in scientific careers. In 1957, when I was just starting my work at college, the Soviet Union released Sputnik, which scared Americans deeply and inspired our then president, Dwight Eisenhower, to launch a major effort to improve science in our country and improve science education. Then during the Vietnam War, there was an opportunity such as the one I had to get trained in science. And then shortly after I moved to San Francisco to start working with Mike Bishop, President Richard Nixon signed the National Cancer Act, which provided tremendous increases in funding of cancer research. Now, in starting out to pursue these goals of identifying cancer genes and understanding how cancer arises, I had to think through what questions I'd be asking. I've told you that the essence of the message that I took away from my experience learning to do science at the NIH was how to define a question that was interesting and answerable and would lead to further questions. And as I thought about choosing questions to work with these cancer-causing retroviruses, at least three came to mind. The first was the question of how retroviruses multiply. Many of you may know that retroviruses have RNA genomes, but when they enter cells, there was reason to believe that the RNA was copied to make DNA. This was an idea promulgated by Howard Temin, about whom I'll say an additional word in just a moment. And he had proposed that the way that is done is by using an enzyme not yet discovered, the enzyme which we now call reverse transcriptase, to make a DNA copy of the viral genome, which could be integrated into the host genome. And that's how retroviruses could perpetuate their cancer-causing information in the chromosomes of host cells. The second question was how do retroviruses cause cancers? There were many ideas that were around. The one I found most attractive and turned out to be correct is the idea that retroviruses carry their own gene, a gene that might or might not be important for replication of the virus, but was responsible for conferring a cancer-like phenotype on an infected cell to turn it into a cancer cell. And the third was a much bigger question, namely, could we learn from our efforts to understand retroviruses how a normal cell became a cancer cell? That problem, exemplified here by a couple of pictures, was really at the heart of all cancer research, and to a certain extent remains at the heart of that research. That is, how does a normal cell, for example, an epithelial cell or a mesenchymal cell or a hematopoietic cell, get converted into a cell which has many altered properties that we often now refer to as the hallmarks of cancer, the ability to grow inappropriately, to evade cell death signals, to invade locally, to grow at metastatic sites, to generate the development of blood vessels that keep cancer cells alive. And it seemed to me that it was possible by studying a virus class like the retroviruses that we might get some clues into how this all happens. And I think you'll agree by the end that we've learned a fair amount about that, but as in all cases of interesting science, the interesting answers we've gotten to these initial questions have only led to other, deeper, better, even more exciting questions. Now, before getting started in describing the work that Mike Bishop and I and many of our colleagues did in the early years in the 1970s, it's worth saying something about what happened before then, because there were many pieces of evidence that suggested that something about genetic change is related to the origins of cancer. And one of those lines of evidence was epidemiological, that is, associations between certain kinds of environmental exposures that were known to cause mutations as measured in fruit flies and cells and bacteria and other organisms. And that included exposure to tobacco smoke, most closely linked with lung cancer, but with many other cancers as well. Exposures to certain industrial substances like asbestos, exposure often just exposure to radiologists from X-radiation that was used in diagnosis and treatment and associated with the appearance of leukemias in the radiologists. Those were suggestions that mutations had something to do with cancer. There was also work done by medical geneticists who recognized that certain kinds of cancers, such as retinoblastoma, certain forms of colon cancer, certain forms of uterine cancer, occurred in families, suggesting that genetics, again, was important. The transmission of variant copies of genes might play a role in initiation of cancer. And the third thing was the thing that was motivating me most profoundly, which is the idea that there were many kinds of viruses, retroviruses like Rous sarcoma virus, the mouse mammary tumor virus, the murine leukemia virus, and some DNA viruses such as the papilloma virus, I'll talk about again a little bit later, simian virus 40, were all viral particles that contained small number of genes, and yet by entering cells and contributing those genes to cells, seemed able to change a normal cell into a cancer cell. And the open question is whether this is some anomalous feature that has nothing to do with human cancer, or whether understanding how a retrovirus or a DNA tumor virus could cause cancer would be revealing about the origins of human cancer. So, let me say a few things about one of these elements, namely the element highlighted in red, the Rous sarcoma virus. Now, Rous sarcoma virus has a very long history, much longer than my own, perhaps not as old as Saha's, but in 1910, an upstate poultry farmer in New York brought to a young physician named Peyton Rous at the Rockefeller Institute a chicken that had a large growth of mesenchymal tissue called a sarcoma, a malignant neoplasm that obviously was on its way to killing this chicken. And Rous had the idea that a virus particle, viruses had just been discovered several years earlier as particles that could pass through a narrow filter and still convey some new trait to a cell or an animal to which it was exposed. So Rous did the following experiment. He took this chicken with a sarcoma, he cut out the sarcoma, he ground it up, he passed the extract through a filter, he made the assumption that the filtrate might have an infectious element that he would call a filterable agent or a virus, and he injected solution containing that material into a new chicken and caused a tumor. And you could do that repeatedly. You could even take that fluid and pass it into certain kinds of rodents and get tumors, suggesting that there was some kind of agent that would multiply when the tumor was growing that would cause a cancer in a new animal into which this filterable agent was injected. Now, that was interesting, stimulating. It didn't allow any rigorous quantitative studies of this element or any concise or revealing genetic approaches. And a major advance occurred nearly 50 years later when Howard Temin, the first person to propose that retroviruses grew through DNA intermediates, and his then mentor, Harry Rubin, then at Caltech, made a huge advance in the study of Rous sarcoma virus by developing a reliable in vitro cell culture-based assay. And the way that worked is that they took a chicken embryo, cut it up, put the cells from the embryo, mainly fibroblasts, into a culture dish, and then applied Rous sarcoma virus, shown here as in a picture from an electron microscope, onto the culture. And every time a single infectious particle and entered a cell. It generated cells that have an altered appearance, as I hope you can see from the back, much more refractal, rounded up, able to grow on top of each other to form a focus of so-called transformed cells, which, when injected into a chicken, formed a tumor. So suddenly we had a quantitative, reproducible system to assay the number of infectious Rous sarcoma virus particles that were able to induce tumors in animals. And that meant you could do biochemistry, you could do quantitative virology, you could do genetic analyses of the virus. And the virus was assumed based on electron micrographic pictures of its genome to have perhaps half a dozen genes, maybe even fewer. Many folks were trying to develop genetic assays that would lead to the isolation of mutants that could be revealing about the functions of the virus. So of all those genetic experiments, the one that I think in retrospect is the most important was an experiment carried out by a postdoctoral fellow, Steve Martin, working then at the University of California, Berkeley. He made a so-called temperature-sensitive mutant of Rous sarcoma virus. And the way that virus behaves is shown in this diagram. The basic idea is that if you infected a normal chicken cell with this virus at a relatively low temperature, 35 degrees centigrade, the virus would do its normal thing. It would infect the cell, change the behavior of the cell so it looked like a cancer cell. The virus would multiply and make many particles. If you did the same thing at a higher temperature, the virus would infect the cell, make many kinds of many particles, but the cell remained a normal fibroblast. If you took the cell you originally infected at the lower temperature and raised the temperature, virus production continued, but the cell returned to a normal appearance. If you went in the other direction, from the higher temperature to the lower temperature, virus production continued, but the cell resumed its transformed appearance. What that suggested that there was some function of a gene that had been mutated in the course of this experiment, that viral gene that was required to continue to make its protein product to maintain the transformed state. Because the protein product was mutant, it was unstable at higher temperatures, and when the temperature was raised, the function of that protein was disabled and the cell went back to a normal appearance. This proves to be a pivotal experiment that changed thinking about genes that might be involved in cancer, because it suggested those genes didn't simply trigger a cancer, but were required to maintain the cancer phenotype, the cancer behavior, and that could lead ultimately, if you were smart and lucky, to the development of drugs that would interfere with the function of a cancer gene product and allow a novel kind of targeted therapy for cancer. And as we'll see in just a minute, that in fact happened. Now, to make greater progress against this form of cancer and to understand the reason why the Rous sarcoma virus actually had a gene involved in transformation, it was necessary to build a genetic map of the viral genome. One of the things you may have appreciated from the previous slide is the transforming gene didn't seem to be required for replication. And indeed, as this genetic map was being built by a number of techniques I won't have time to describe, it became apparent that there were three genes, or roughly three genes in the viral genome that were dedicated to the multiplication of the virus. A gene for making the core proteins of the virus, a gene for making the enzymes, especially reverse transcriptase, but also a gene for integrating viral DNA into cells and for chopping up proteins that were required for making a viral shell, and then a gene that was required to make envelope proteins that would be on the surface of the virus particle. And in addition to those genes required for replication, there was a fourth genetic element that became called SARC, or VSARC for viral SARC, SARC standing for sarcoma. That was the gene that we've been talking about in in thinking about Steve Martin's experiments. Now, that gene was characterized by a lot of point mutations of the kind I've just described, but also deletion mutations that afforded an important opportunity, especially in the age before recombinant DNA techniques, to make a radioactive probe, a piece of DNA that was specifically missing, or I should perhaps put another way, that specifically represented the region of the Rous sarcoma virus genome that was deleted to make Rous sarcoma viruses that were defective in transformation. And it was by using the radioactive DNA that was specific for the Sarc gene that my colleagues and I were able to show that the Rous sarcoma virus gene was not like other viral genes. It was clearly derived from a normal host gene that, like other cellular genes, was composed of introns and exons, highly conserved in evolution, and was somehow, we think we know how, but I don't have time to describe that, somehow acquired by precursors to Rous sarcoma virus that had the genes for replication of the virus, but didn't have an element that would mediate transformation of cells by the virus. So the viral Sarc gene derived from a normal cellular gene, but as you might expect, since chickens normally don't have tumors, that gene was slightly different in a way that made it unable to produce tumors without subsequent mutational steps. So in other words, one way to summarize this is to say that the Rous sarcoma virus RNA genome, shown near the top of the slide, had a region we call the viral Sarc gene. That encodes a protein of about 60,000 Daltons that we call the VSarc protein. And the normal cellular gene has a very similar sequence, but not identical. It makes a similar but not identical protein. And once it was possible to show that this protein was in fact an enzyme which has some very interesting properties. It transfers phosphate from ATP to specific amino acids in proteins. And unlike most so-called kinases in cells, it transfers phosphate not to serine and threonine, but to tyrosine, making it a member of a special class of tyrosine, of protein kinases known as protein tyrosine kinases. And as you might deduce from what I just said, the Sarc protein made from the viral gene is very active as a result of mutations that change the way in which the protein gets regulated by other events in the cell. And the cellular gene product has a very low activity, which is sufficient for carrying out the normal functions of the cellular Sarc gene, but is not by itself a transforming gene. So this is a good place to pause for a moment and review a couple of things I've told you about Rous sarcoma virus. That the virus carries a cancer gene, the VSarc gene, and that gene encodes an interesting enzyme, a protein tyrosine kinase. That gene was somehow derived from, stolen from, a normal cellular gene, highly conserved in evolution, which makes a protein that's similar to, but slightly different from, the viral transforming gene. And now I'm going to tell you something in addition, which is that those other cancer-causing retroviruses that I've ignored until now also carry cancer genes that can be defined by genetic mutations. And those genes are also derived from normal cellular genes. Here is a somewhat amusing cartoon drawn by Jamie Simons at the Salk Institute over 30 years ago, showing that in general, retroviruses acquire cellular genes as they pass through mammalian cells, and that those viruses have a variety of genes with a variety of names, and each of those genes was derived from a normal cellular gene. Some of those are highlighted here, such as the ABL gene and the MYC gene and the RAS gene, because they are extremely important in human carcinogenesis, as shown by a number of the figures of folks who were shown in the bottom part of this cartoon. So we need to add to our list of what we've learned the fact that mutations in these cellular genes drive many human cancers. And some of those cancers can be treated with drugs that are targeted against the mutant proteins. I'm going to illustrate this in a couple of ways, but suffice to say that overall, something in the order of 5 to 10% of human cancers now get treated by so-called targeted therapies that are directed specifically to mutant genes that have undergone mutations that drive cancerous development in human beings. And many of those genes are genes that we first learned about long before the Human Genome Project, long before we had DNA sequencing, even before we had recombinant DNA technologies, through the use of retroviruses that carry cancer genes and transform cells. The classical example of all this is the situation in a disease called chronic myeloid leukemia. That disease was described about 150 years ago, probably about the time of Saha's birth, and it was shown to be characterized from experiments done about 1960 by the appearance of an abnormal chromosome called the Philadelphia chromosome, which turns out to be an amalgam formed of most of chromosome 22 and a little piece of chromosome 9. The fusion between those two chromosomes is always almost exactly at the same place, and the result is to join part of a gene known as the ABL gene, the precursor to the viral ABL oncogene, with another gene of much less interest that nevertheless allows the fusion product to be poorly regulated so that it has an uncontrolled activity that's capable of turning the precursor to myeloid blood cells into cancer cells. And that allows the ABL kinase present in that fusion protein to be a target for drug therapy. And indeed, as I suspect many in this audience know, a drug called imatinib, commercial name is Gleevec, was found to be a profound inhibitor of the BCR-ABL fusion kinase protein that is responsible for causing chronic myeloid leukemia. This was found by a rather circuitous route I don't have time to describe, but a kind of unexpected collaboration between drug companies and academic scientists at the Dana-Farber Cancer Center. It was found to inhibit 3 major tyrosine kinases, the ABL kinase and a couple of others, and when it was first placed into clinical trials after work with mouse models of this disease showed how effective it was, and virtually every patient in the phase 1 trial, which was designed primarily to show the safety of the drug, went into a profound remission, resulting quite quickly in approval by the FDA of this drug for use. And now, thanks to this drug and to a few related compounds that also inhibit the ABL kinase, patients who have this form of leukemia, which was formerly a death sentence, usually death occurring within 5 years after diagnosis, now have a normal life expectancy. Moreover, it was found that some of the other kinases inhibited by this drug would allow the drug to be used for the treatment of those diseases, which included some other forms of rare leukemia and a solid tumor known as gastrointestinal tumors, of sarcomas of the gastrointestinal tract that have mutations in either the kinase known as KIT or a kinase known as the PDGF receptor, which are frequently mutated in that disease. So this was a huge triumph, and of course it inspired a great deal more effort to identify activated cellular genes, mutated cellular genes that made proteins that could be inhibited by drug therapies. And this, in a sense, was the beginning of a modern era in cancer genetics, because by this time we did have recombinant DNA technologies, we did have efficient sequencing tools. We did have access to the human genome through the Human Genome Project. We did have a lot more knowledge about what genes were mutated in which kinds of cancer and what kind of proteins those genes might encode. So one of the things that became immediately apparent is that most cancers have multiple mutations, many kinds, base substitutions, termination codons, rearrangements of various kinds, gene amplification, and that some of those genes that were altered by mutation could be targeted by drugs like Gleevec. A very small catalog is shown here with Gleevec at the top, or chronic myeloid leukemia at the top, followed by breast cancer with a couple of examples, and lung cancer with a couple of examples. I'll show you more in a moment. Melanoma, which frequently has a mutation in a gene known as BRAF. And all of these can be inhibited by antibodies and more commonly by small chemical compounds or drugs. When one looks carefully at lung adenocarcinoma, the most common form of lung cancer, which is the most common cause of death from cancer worldwide, we see that while a quarter of the cancers have no obvious dominant mutation, and another third or so have mutations in a very important gene known as the RAS gene, for which we don't yet have therapies, most of the other cancers, nearly 45%, have mutations in genes that encode enzymes for which inhibitors do exist. And those inhibitors are listed on your left, and these are all in clinical use, so that adenocarcinoma of the lung is now a disease for which it's reasonably likely that there will be a drug that will produce at least a temporary remission. These drugs are not cures. You need to keep using them, and most patients within 1 year, 5 years, or 10 years have developed drug resistance, which requires either a different drug or another kind of approach to therapy, like radiation therapy. So we haven't cured these diseases, but we've made remarkable progress against them. So this means that that the modern era is coming into play because of these targeted drugs, but it's also true that mutations that are driving cancers are generating mutant proteins that might be recognized by the immune system. For over 100 years, people have been interested in the idea that the immune system, which apparently exists to deal with the entry of foreign material into our body, viruses, bacteria, and so forth, might also be instrumental in controlling cancers, because cancers might have abnormalities that are viewed as foreign by the immune system. And indeed, it's obvious that if we have multiple mutations in virtually all cancers, that some of those mutated proteins are likely to be perceived as foreign by the immune system. Yet, in most of the time in which it seemed reasonable to assume that the immune system could be a deterrent to the growth of cancers, we've been extremely unsuccessful in trying to exploit the immune system to protect ourselves against cancers. Nevertheless, in just the last several years, last decade or two, some new approaches have been used to make immunotherapy an essential feature of the modern era in cancer care and cancer genetics. And I'm going to tell you just a couple of things about that. None of this is my own work, but it's nevertheless related to the whole idea that mutational changes in cancers are instrumental to the process of generating this disease, and also fundamental to the strategies for treating it. So we now have quite a few approaches that are immunologically based to controlling cancers. The first is perhaps the simplest. That's the idea of using antibodies, especially monoclonal antibodies, directed to proteins that are found on the cell surface of cancers. The one you probably have heard about is Herceptin, which is directed to killing breast cancers that have high levels of a protein known as HER2 on the surface. The second is the use of genetically altered T cells, a major component of the immune system, to attack cancer cells that have unusual proteins or even normal proteins present in great abundance on the cell surface. And the third approach, the one that is most widely used and is in the minds of most people these days, because this year's Nobel Prize in Physiology and Medicine is being awarded to Jim Allison and Tanjo Hanjo, who developed the strategies for trying to enhance the ability of an immune system to recognize cancer cells by taking advantage of the fact that immune systems are not simply designed to convert normal immune cells to cells that are activated against a foreign body or a cancer cell, but they're also influenced dramatically by breaks, by negative inhibitory factors that constrain immune responses. And that's obviously important because one wants to avoid responses against self, and autoimmune diseases are one way of viewing an aberration in the immune system that happily are rather infrequent. But the work of Hanjo and others, and Jim Allison, have revealed the presence of cell surface proteins in immune cells that are the targets of inhibitory forces that restrain the activity of T cells. And by using antibodies against those cell surface proteins, it's possible to release some of those restraints and make the immune system more active in confronting cells that are perceived as abnormal, namely cancer cells that are making mutant proteins. So just to summarize briefly where we stand with all this, there are now several types of tumors, most notably melanomas, in which many of the first experiments were done, and some leukemias, but also lung, renal, bladder, colon cancers can be treated with the antibodies that interfere with the checkpoints that exist in the immune system. The efficacy of these antibodies has been increased recently by using combinations of those antibodies, and there's a good prospect for increasing the efficacy yet further in trials that are examining whether these antibodies that relieve inhibition of the immune system can be combined with either targeted drugs, conventional chemotherapy, or radiotherapy. We still don't know exactly which antigens, often newly created antigens from mutant genes, are the actual targets for the immune response, but there is a tremendous amount of work underway. And I hope some of you will appreciate the irony of this situation, namely that mutations which are perceived as the cause of cancer are also responsible for generating the susceptibility of certain cancers to these new forms of immunotherapy known as checkpoint blockades. So one of the indications for using this new immunotherapy is the presence of high burdens of mutations in certain cancers. Cancers that have, for example, a defect in DNA repair systems are frequently the most responsive to these new immune therapies. Finally, let me just say a couple of words to round out your view of the modern era in cancer genomics about how we now view the extensive mutational activity that can be found in cancer cells, so that we begin to think of cancers not simply as simple diseases caused by one or two mutant proteins, but instead as Darwinian systems of evolution that occur over fairly short periods of time, with random mutations leading to cancer, random mutations continuing to occur, with continued selection for what we would call fitness, an ability to grow with a special avidity, an ability to invade and grow in distant sites, an ability to evade the effects of various kinds of drug therapy, and the substrate, the patterns of mutations that allow such evolutionary change to occur can be appreciated by just simply by looking at one of several data sets generated over the last 10 or 15 years by using high-throughput genome sequencing to characterize cancer genomes. So in this particular experiment carried out by Matt Meyerson and his colleagues, roughly 230 lung adenocarcinomas in human beings, represented along the horizontal axis, are shown in relation to genes that are repeatedly mutated in these cancers. And the things you can see is that the genetic pattern is highly heterogeneous. Some genes are mutated fairly frequently, some much less frequently, but the patterns of mutation are so varied that there's no single tumor that has the identical set of mutations. And furthermore, each of these occurs in an individual who has many other variations that distinguish him or her from other patients. Now, selection patterns for this ongoing process of mutation gives rise to evolutionary charts that are not unlike the kinds of things that Darwin drew in the very early days of his thinking about evolution. Just one simple example here from the work of my colleague Charles Swanton in England shows the evolutionary tree with successive subclones arising from a single case of renal cancer, not unlike what Darwin put in his notebook 150 years ago, showing that single original life forms might evolve into a variety of species as a result of random mutation and selection. And his positing, I think, about that phenomenon of how the diversity of life is generated is closely similar to the kinds of patterns we can now draw when we take multiple samples from a single patient with cancer and try to recreate an evolutionary tree for those cancers. Now, much more could be said about the properties of cancers that are undergoing this extensive mutational process, but I want to turn my attention now to a more social phenomenon, and that can be stated simply here that we have a lot of new knowledge about the genetic basis of cancer, and that is changing the kinds of questions that we are able to ask experimentally. There are several examples of that under study in my lab and others, and I thought I'd just illustrate one to whet your curiosity about how experimental approaches have been changed by what we've learned. And I'm going to look at just briefly at the notion of how we begin to investigate why certain kinds of cancer have the genetic abnormalities that they generally have. Those changes may be very heterogeneous, but there are commonalities that are very striking. Lung cancers of certain kinds having one general pattern of mutation, other kinds of lung cancers having another. So I remind you that we started this discourse together by looking at the first cell culture-based means of assaying the oncogenic effects of Rous sarcoma virus. It's now possible to do such work not with another animal species, not with chickens, but with humans, to use cell types that are much more commonly involved in cancer than fibroblasts, and by avoiding the use of viruses and just by taking altered genes and introducing gene alterations directly into cells. My own lab has been working on this problem in the study of a highly lethal cancer known as small cell lung cancer, disease that causes about 30,000 deaths a year in the US every year. Virtually every patient is a smoker, but these cancers have fairly similar patterns of mutations. There are variations, there is heterogeneity, but virtually every cancer has the following properties. First, they express the markers that are associated with a certain rare cell type in the lung, known as a neuroendocrine cell, and they almost always have show loss of a set of genes known as tumor suppressor genes that I haven't had time to discuss here. But what we do in these experiments is to take cell lines derived from human embryos that have been donated to research, to grow those cells in tissue culture, to carry out a differentiation process with a formula developed by several lung developmental biologists to make a variety of cell types, to then do something novel, the work of a postdoc in my lab named Joyce Chen, to block signaling through the Notch signaling receptor, to induce significant numbers of lung neuroendocrine cells, and then by inactivating certain tumor suppressor genes known as the retinoblastoma gene and the p53 gene, we can make these cells form tumors that resemble morphologically small cell lung cancers when injected into the subcutaneous tissue of a mouse. So this illustrates the ability of our ability with new knowledge to do a transformation assay that is fundamentally similar to what was developed 50 years ago, but to do so with human cells using genes that we know are involved in the generation of those tumors, and to begin to make tumors of specialized types that are responsible for large numbers of human deaths, and to do this in a way that hopefully will reveal the vulnerabilities in the generation of tumors that can be addressed more effectively in our efforts to treat this uniformly lethal disease. So that brings us to a new point to step back a second to say that cancer research has gotten rather sophisticated. Cancer treatments are not only sophisticated but also expensive, and we have to ask ourselves whether we're doing enough to ensuring the benefits of research to all populations, to everyone on the globe. We know that we've been reducing the death rate from cancer in the US as a percentage of the population by about 1.5% a year for the last 20 or 30 years. Most of that, or at least half of it, is due to reduction in smoking, but much of it is due to improved cancer therapy. And we're proud of that progress, but we wish it had been more dramatic. But as we look out at the future with people living longer and healthier lives in poor countries, we can see that as the populations age, that the number of deaths that will be attributable to cancer with time will increase dramatically, especially in low and middle income countries, with the death rates from cancer in the advanced economies showing very little change. And that has given many of us a moment of reflection about what we're going to do to try to mute or reverse this anticipated increase in the burden of disease in developing countries. When I was director of the National Cancer Institute in the first 5 years of this decade, I assembled a new center for global health to try to confront some of these problems, and they're not easy to confront because every country has a different set of cancers that are common. Countries have very different levels of receptivity to efforts to control cancer because of their attitude toward the commercialization of tobacco, or their existing resources for health care, or because of their research capabilities. And we recognize that we can't just treat our way out of this problem, that we'd have to make a much more complex approach that includes prevention, early detection, as well as treatment if we're going to make progress. So we established this center in 2011, headed then by a long-standing member of the NCI staff, and the goals were fivefold. One was to help countries develop national cancer plans. The second was to try to take advantage of the higher frequency of infectious causes of cancer in certain countries by using existing vaccines or trying to develop new ones, to improve the treatment of healthcare overall, improving surgery, improving healthcare systems, finding ways to palliate symptoms and the often horrible death and pain that accompany the end stages of cancer by trying to take advantage of situations where changes in behaviors, like the use of tobacco, obesity, use of alcohol, which are instrumental in controlling other diseases, could also be applied for the benefit of reduction of cancer incidence, and then to try to harness the considerable enthusiasm we find for improving global health among students, especially medical students in the US and elsewhere. I would just like to make one brief detour before in dealing with this problem of global health by talking briefly about oncogenic infections. And I've mentioned already that there are viral agents that cause or are associated with cancers, and one of those is the human papillomavirus, or HPV, highlighted in this slide. The other virus, hepatitis B virus, is also a virus for which good vaccines exist. And I want to say a word about human papillomavirus vaccine because it is a particular conundrum here in India. Worldwide, human papillomaviruses, of which there are many strains, are responsible for hundreds of thousands of deaths from cervical cancer, but they also contribute appreciably to other forms of cancer: anal cancer, vulvar cancer, cancer of the penis, mouth and head and neck cancers, and the opportunity to use a vaccine to prevent the infections that cause cancer seem particularly appealing. So there are a number of things one has to know in thinking about the application of these vaccines to the problem of cervical cancer and other HPV-related cancers. There are many strains of this virus, but only a few are highly cancer-causing, and therefore it's been possible to make vaccines against just a few of those strains that allow over 90% protection against infection by those cancer-causing strains. It's now possible to reduce the recommended number of doses of the vaccine from 3 to 2. The benefits are clear. It's protection against cancer. It's protection against cancer mortality, especially in women who are childbearing, and it relieves the orphancy that follows the loss of life in women who are bringing up children. And there are a number of issues. The vaccine, as currently marketed, is not cheap. The price can be reduced by selection by the Global Alliance for Vaccination and Immunization that will help support the cost of these vaccines. There's argument about when people should receive the vaccines. Some parents feel that to receive this vaccine is an admission that the child will have some sexual activity at some time in their lives. That usually does happen, but to barter away the notion that the child is not going to have any sexual activity for vulnerability to infection with a cancer-causing virus seems like an easy argument to deal with. There is, because the viruses are not totally protective, at least in the US, there's continued need for pap smear as a screening device, and for a number of reasons having to do with cost and association with sexual activity in the transmission process, the uptake has been slow even in the US. Certain countries we've been incredibly successful. In Rwanda, the government has cooperated with drug companies and with Gavi to make vaccination virtually universal. In India, things have been much more problematic. There is a high toll of cervical cancer, yet adoption of HPV vaccination has been very slow for a variety of reasons. One is the obvious one, cost. The other is a non-obvious reason. There was a trial that was, in my view, misinterpreted but was carried out without adequate consent by parents of the children who were vaccinated, and there remain limited funds from the public sector to support the cost of the vaccine. However, I was encouraged last year when I was visiting North India and heard a report from the Punjab about a demonstration project in which the cost of the vaccine was curtailed by support from the Global Alliance, and parents were adequately informed. There were no claims of side effects, and I believe that a more robust vaccination program is now possible. Okay, those are the main things I wanted to say. I would like to conclude, though, with a few remarks about the conduct of science and what it takes to make a successful scientific enterprise, in the spirit of telling you something historical about my own conversion from an aspiring medic to a scientist. I've given a lot of thought over the years during my time as a leader at the NIH and as a leader of a cancer center and as a general commentator on the sociology of science. And I'd like just to conclude with a few remarks with some illustrations from my own experience of what it takes to make science a successful enterprise. The first thing is creating teams and partnerships and developing mechanisms for people to feel rewarded about successful science, even when they're working not alone and independently, as is commonly portrayed in the literature about science, but instead as members of teams and partnerships. In my own life as a scientist has been deeply crafted and influenced by a long and productive relationship with my colleague Mike Bishop, with the large team of postdocs and students who've worked with us, and that has always been, to me, emblematic of the importance of working collegially with fellow scientists, making use of disparate skills and multidisciplinary teams, and increasingly, as we need to accumulate information about the vast array of diseases we lump together under the term cancer, it's become important to examine that data and gather it and take advantage of it through the use of large teams in which it's impossible to give credit to one or two people. That credit simply has to be shared, and we need to have ways to recognize contributions that can't be simply deciphered by the position that an individual has in a long list of authors. The second thing I want to talk about is political and financial support. Science costs money, and science in our country and yours generally comes from the government, and having governments that are committed to the support of science is highly critical. In the US, we've been fortunate. Our founding fathers were members of the Enlightenment. In the middle of the Civil War, Abraham Lincoln took time out to found the National Academy of Sciences. He also built a transcontinental railroad and started land-grant colleges that emphasized the agricultural sciences. This was his miracle year, but it reflected his commitment to science. The National Institutes of Health, where I've spent a lot of time and been deeply influenced, it has been critical to have our national leaders strongly supportive of what we do. One of the first to show that support was Franklin Roosevelt, who took time out from his presidential campaign in 1940, when we were at the verge of World War II, to dedicate the new NIH campus. It's exemplified by Harry Truman, his successor, who went to the NIH to lay the groundstones for the clinical center that has characterized the nature of clinical research in the country, specifically at the NIH. And most recently, not necessarily today, but fairly recently, we've had the support of Barack Obama, who had hired me for 2 separate jobs, shown here next to a model of the double helix, announcing an initiative in precision medicine. The third point I want to make has to do with sharing information in an open fashion with our colleagues and with the public that supports us. I like looking at this picture by Rembrandt, of Dr. Tulp, who is exhibiting for the burghers of towns that surrounded Amsterdam in the early 17th century, his knowledge of human anatomy, and he seemed eager to share that information with his colleagues, but I'm not convinced that we as scientists are always quite so eager to do so. So that suggests to me that we have a kind of paradox. We, the scientific community, developed the internet, which enables rapid and immediate and nearly cost-free sharing of our information, but at least in biology, we have been very slow to take advantage of the internet to improve scientific publishing and communication. And in the late 90s, when I was director of NIH, I began to try to influence my colleagues to change the way biomedical research is communicated. One way to pose the question is whether we're going to put our information into a monk's study and shield it from the public, or whether we're going to put it into a public library like the New York Public Library shown on our left. And there now appears to be at least 3 ways to be open about what we've learned. One is to create public digital libraries like PubMed Central at the NIH, which allows public sharing of information once the journals have agreed to allow it to be open. And at the NIH now, you cannot get your grant renewed if you don't have your papers available to the public through PubMed Central within a year of publication. A second way to proceed is exemplified by what's called open access publishing, a form of publishing in which the business model is fundamentally altered so that authors using money from their granting agencies pay a relatively small amount to allow the work to be seen digitally for free by anybody in the world at the time of publication. And the third is to make access even quicker because the review process is frequently slow by allowing articles that have been written but not yet reviewed, so-called preprints, readily available. And that could extend not just to manuscripts but also to data that have not yet been assembled into manuscripts, but nevertheless could be shared by colleagues who have an interest in promoting those features. Now, open access publishing has been controversial, but to me there's no controversy. It's almost self-evident that all of science and the public that supports it would benefit from a system where articles are made free via the internet at the time of publication with a great savings of cost. Currently, the subscription-based journals make great profits for their stockholders that are unfortunately limiting to the scientific community because they don't allow everyone to read the papers, and you can't read the papers until you pay a subscription fee, and that is no longer, in my view, necessary. Now, because of a variety of efforts, the creation of Public Library of Science, the willingness of certain journals to become open access journals, the creation of a number of other open access journals, the pressures that funders of science and the public are placing on scientists to make their work more easily shared. At least 30% of the world's literature, at least European and American literature, is made available at the time of publication. I believe that actions by funders will make that an even higher number soon. And then there's the advent of preprints. Preprints have been used in physics, as I'm sure many of those at the Saha Institute know, since 1991, thanks to the activity of my friend Paul Ginsberg, who was then at Los Alamos, now is at Cornell, who created a repository known as the Archive. Now, thanks in part to the advocacy by my former UCSF colleague Ryan Vail, much more of the biological literature is being placed into preprint form, largely in a preprint server known as bioRxiv, managed by Cold Spring Harbor. So we're all excited by this. I put all of my new papers right into bioarchive even before they're reviewed, to be sure that I'm sharing what I've learned with my colleagues as quickly as possible. Next to final point is the need for international work in science. Science is a universal, a global activity. When I hear my new president talking about America first, I cringe because there is no reason, and there are many reasons why, against making science a product of nationalism. All of us who run laboratories of moderate size know that our students and postdocs and colleagues come from everywhere. Over a quarter of the U.S. National Academy of Sciences are people who were born abroad. This is a profile of my own laboratory in 2015 when I was at Memorial Sloan Kettering, posed on one winter's day in Central Park. You can see the Gates art project, the orange banners shown behind, and you can see people from many different countries, including India, but also Russia and China, Taiwan, other places, gathered together because we work more effectively together. That notion of internationalism applies not just to people who do medical science, but to those whose diseases are being studied. In the cancer domain, we are now in the process of gathering large amounts of information about cancer genomes, but the vast majority of that information comes from the study of patients who are European whites, in the range of 80 to 90% of data in the major databases, some of which are shown here, like the Cancer Genome Atlas database. And that is a limitation because we are learning too little about how the entire human species has received genetic variants that influence the kinds of genetic changes that lead to cancer and how those cancers respond to therapies and how often drug resistance develops. This is a clear limitation in our efforts to understand cancer. So an organization for which I also work, the New York Genome Center, is undertaking a new project called Polyethnic 1000, which is an effort to study cancer genomes throughout New York City, a city rich in what we call minority populations, African Americans and Hispanics, but also rich in immigrants from countries like India and China and other places that unfortunately develop cancer when they're in New York and could contribute dramatically to our knowledge about the genetic variations that predispose or protect from cancer over the years. So what I've tried to tell you about is the many ways in which we're trying to confront cancer, the progress that's been made over the last 50 years or so, and some of the rules by which I believe progress can be made more rapidly by paying attention to the sociological and political features of our landscape that have a heavy influence in how well we're going to do in this quest to control a dread disease. Thank you again for inviting me here for this memorial occasion and a chance to honor Dr. Saha. Thank you.
Thank you, Professor Varmus, for the amazing talk. And since this is a memorial lecture, we usually do not have questions, but in case you insist, we could have one or two of them.
Well, I won't insist on anything, but I would welcome questions, and
Yes.
I don't see why a memorial means we don't have questions. I hope when I'm dead, there will be questions asked anyway.
Definitely. So we definitely have time for 2 or 3 questions. There is a question up there. Yes. Please switch it on. ওটা অন করে দিন একটু. Thank you.
Yeah. question is that in spite of the newer developments on chemotherapy, in your regime, for example, the antibodies and the chemicals that you developed, and also the most recent development by these two Nobel laureates this year, 2018 Nobel laureates, where the immunotherapy is coming for the treatment of cancer. Why then are you predicting that the number of deaths due to cancer is going to increase more and more, in spite of the availability of the new these therapeutic agents?
Okay, that's a good question. So the question is, I've been optimistic, and I certainly have lauded the developments in immunotherapy. You're absolutely right, and you might think that if I sound optimistic about or proud of the fact that the scientific community has developed these immunotherapies, why shouldn't I be predicting that deaths from cancer are going to decline? Well, okay, there are a couple of explanations. One is completely trivial, and that is that those slides were made based on World Health Organization data before the immune therapies were developed. That's a trivial answer. I would still predict, and I want to check with my colleagues, but I would predict that those numbers are not going to be much affected in the foreseeable future by the immunotherapies. First of all, the immunotherapies are only used in a fairly small subset of patients, perhaps 10 to 15%. Some of those do remarkably well and have long-term remissions that may last a lifetime, but many of the responses don't last more than a few years, and some patients experience significant toxicity from those therapies. The next point is that the increase that I was alluding to is going to occur largely in developing countries. Even in the U.S., not everybody has access to these very expensive drugs. And over the next 5 or 10 years, 15, even 20 years, as new forms of these therapies come into practice, but protected by patent rights, they're going to be still limited in use because of the cost of these drugs. I would suspect, I don't know the numbers, but I would wager that in India, very few patients are receiving these new immunotherapies. I know that some of the targeted drugs are being used, and of course some of the targeted drugs are coming off patent and their prices will decline very rapidly. But I'm glad you asked the question, because there is a fundamental inequity worldwide with respect to how patients with cancer are treated. Some of those are not of ill intent, we don't have healthcare systems that adapt as quickly as I would like to changes in cancer therapy. Then there is what I would consider to be an inequity in which wealthy people in many countries, even in developing countries, have high degree of preference for access to these novel, often powerful, but expensive therapies.
DNA topoisomerases are the targets of antibacterial, antiparasitic, anti-cancer, antiviral agents. So, what is the present status of camtothecin, which is also considered to be the very good inhibitors of DNA topoisomerase 1b, and also a very good anti-cancer agent. There are so many compounds which are derived from this camtothecin, which may come to the market. What is the promise?
So you're asking a question about the use of topoisomerase inhibitors in cancer therapy, is that?
Yeah.
Yes. So there are a number of topoisomerase inhibitors, and I'm glad you asked that question, because when I refer to chemotherapy and distinguish it from targeted therapy, some people say, well, a drug that's directed against a DNA polymerase or a topoisomerase or a drug that interferes with incorporation of nucleotides in the DNA, those are biochemically targeted, and that's true. But they're not biochemically targeted so much based on different mechanisms of cancer in different cells. The use of targeted therapy, as we currently use it, is specific for the identification of a genetic abnormality that can be measured by DNA sequencing and other forms of DNA analysis. Topoisomerase inhibitors do have a role in the treatment of cancers, especially lung cancer, for which the new targeted therapies cannot be applied because we don't have a mutation that corresponds to one of the new drugs. Those drugs have been successful in prolonging life. They are not cures for cancer. They have some toxicity. I'm not sufficiently familiar with the use of those drugs. I'm not a practicing oncologist, but they certainly have not been highly effective agents of the kind that have been seen in the treatment, for example, of lung cancer with erlotinib, where patients often go into complete remissions for many years without any severe toxicities.
So we'll have time for one last question before we begin the next.
Can it be a woman? We haven't had a woman
Yes,
ask a
there is
question
one
yet.
lady up there.
Yeah.
There is one lady up there
Okay,
who
we'll
wishes to ask.
All right. So
Please stand up.
We can take yours as well, and then we'll take.
Yeah.
Okay. Ladies first.
Sorry. Hello. Targeted therapy by using drugs like prazopanib or something else, how far it is successful when cancer has reached the metastatic stage?
Well, first of all, targeted therapy is almost always used when cancer is in a metastatic state, because if there's no metastasis, you can usually remove cancers with surgery. And if it wasn't apparent to the entire audience, let me make it apparent that surgery is still, in most cases, the best way to treat cancer. You take the whole thing out and you cure it. And all this excitement about scientifically based therapies is wonderful for people who have no other avenue of treatment. But the best thing to do is to find cancer early before it's metastasized and to remove it and cure it. So almost all patients who receive targeted therapies have metastatic cancer that can't be removed by surgery or treated simply by radiation therapy, which is, again, often curative.
Yeah, but a particular medicine is directed towards a particular cell. For example, prazopanib is for RCC. So if the cancer is spread to liver, etc., how does this targeted therapy will work? For example, it travels from kidney to the liver.
It depends on the nature of the primary cancer, the constellation of mutations that are in that cancer. I think you're asking a question that is interesting and probably hasn't been paid enough attention to, which is the question of whether certain targeted therapies work more or less well when the metastasis that you're trying to treat is in one organ or another.
More than one.
And I don't think there's a simple answer to that question. It's probably an important question because we know that cancers are heavily influenced by what we call the microenvironment, the nest of supportive cells and blood cells and blood vessels and immune cells that are found in any organ in which the cancer is grown. So it's probably a question that requires more attention than I think has been given to it.
Thank you.
Sir, since you have played a leading role in making, socio-political decision-making related to science, my question is, we are talking about cure of science always, but looking at cure of science, like cure of cancer, cure related to cancer. Scientific cure. So my question is, why don't we think of prevention? Take, for instance, tobacco use. what is the overwhelming constraint of putting a blanket ban on tobacco use? We are always talking about what are the clinical costs of tobacco use for the next 10 years.
No, in fact, I appreciate the question, but in a sense it just emphasizes something I did say, which is that, that especially when you start thinking about public health and the global effects of cancer, that it is very important for every country to think about its preventive strategies. And in the agenda for the NCI's Global Health Center, the things that I emphasized were vaccines, tobacco, obesity, alcohol, other social programs, alleviation of symptoms. Prevention is critical. Early screening is important for trying to detect some cancers early before they've spread and they can be surgically removed. So prevention, yes, is critical. The problem we've had in the preventive sphere is that we make very poor use of the tools we already had in hand. I would like you to go out and convince the Indian government to ban tobacco use. Lots of luck. I'm behind you, though.
Sorry, we have to now. We now need to wind up. So I would like to request our director, Professor Ajit Kumar Mohanty, to please come on stage and felicitate our speaker.
That's a wonderful word, felicitate. Sure.
Thank you. Thank you, Professor Mohanty. Thank you, Professor Varmus. And I thank you all in the audience for making this a success. Thank you very much. Please join in for tea outside. Thank you all.